Modern cellular biology rests upon a foundational concept known as the fluid mosaic model. The fluid mosaic model proposes that a cell's outer membrane is a flexible lipid sea where protein molecules float freely. This model operates on a very specific physical assumption about the interior of the cell. It views the cell as an osmotic bag, a semi-permeable lipid barrier holding a dilute, largely unstructured aqueous solution. Viewing the cell as a fluid solution creates an immediate physical problem. According to the simple laws of diffusion, ions suspended in a fluid environment should rapidly cross the semi-permeable membrane until their concentrations equalize on both sides. Yet observed biological reality contradicts this expectation. Living cells maintain steep, highly unbalanced concentration gradients, characterized by a massive volume of internal potassium ions and a strict exclusion of internal sodium ions. To reconcile the fluid model with these observed gradients, standard biology adopted the membrane pump theory. The membrane pump theory posits the existence of constant, energy-consuming active transport mechanisms like the sodium-potassium ATPase pump. The sodium-potassium ATPase pump is a specific protein complex tasked with moving sodium out of the cell and pulling potassium in. This creates a strict dichotomy in how we understand cellular physics. We either accept a model requiring active, continuous transport against physical laws, or a model where an inherent physical structure holds the elements in place. By relying on the membrane pump theory, standard biology dictates that a cell must devote massive, continuous amounts of its metabolic energy strictly to fight diffusion and keep its internal ions from equalizing. In the mid-20th century, Dr. Gilbert Ling initiated a rigorous biophysical critique of the membrane pump theory, challenging the basic energetic math required to keep these pumps running. Ling identified a thermodynamic paradox at the core of the standard model. Perpetually forcing ions up a steep concentration gradient demands an immense, uninterrupted supply of cellular energy in the form of ATP. Ling audited the energy requirements of the sodium pump in frog muscle cells. His calculations revealed that operating the required pumps continuously would consume an amount of energy that vastly exceeds the cell's maximum possible ATP production capacity. Furthermore, Ling noted that conventional physiological measurements frequently underestimated the actual rates of sodium efflux by a factor of at least 10, masking the true severity of the energy deficit required to run the pumps. He proved this empirically. Ling disabled the active pumping mechanisms in cells by cutting off their energy supply, yet observed that the tissues maintained their highly skewed sodium and potassium distributions entirely intact. The academic establishment largely ignored Ling's thermodynamic audit. Institutional grant structures and peer review systems tend to fund research built upon established premises, entrenching the membrane pump theory despite its mathematical impossibilities. The conclusion drawn from Ling's math is straightforward. If the cell physically cannot produce enough energy to operate these hypothetical pumps, the foundational premise of the cell as a fluid osmotic bag is broken, and a new physical model is required. Before replacing the fluid model, the complex internal geography of the cell requires scrutiny. Researcher Harold Hillman provided a methodological critique of how cellular ultrastructure is actually visualized. Biological tissues cannot be placed directly into an electron microscope. The sample must undergo severe physical and chemical interventions, including rapid freezing, saturation with chemical fixatives, and staining with heavy metals. These immense industrial machines require the biological sample to be completely dead, desiccated, and chemically altered before an image can be captured. Based on the solid geometry of these preparations, Hillman concluded that the highly compartmentalized structures we see, like the endoplasmic reticulum and the Golgi body, are likely chemical artifacts, appearing only as precipitations caused by the aggressive fixative agents themselves. By stripping away these artifactual internal compartments, the visual evidence aligns with a different reality. The living cell interior is a holistic, bulk-phase, granular gel system. Gilbert Ling provided the biophysical framework for this gel state, calling it the association-induction hypothesis. The association-induction hypothesis is a framework suggesting that the cell's components are tightly bound together in a highly ordered, solid-state gel. Under this hypothesis, the interior of the cell operates as a highly organized, low-entropy, solid-state system. It is defined by physical structure, not fluid dynamics. The central organizing mechanism is an extended protein matrix spanning the cell. The electrical charge of this extended backbone forces virtually all the water inside the cell to organize into a dynamic, rigid structure called polarized multilayers. Water rigidly structures layer upon layer against the protein. This tight geometry solves diffusion. Large sodium ions physically bounce off, maintaining low internal levels without active pumping. Conversely, smaller potassium ions slip through the lattice, chemically locking into specific receptor sites on the matrix. Ling's theory maps perfectly to modern biophysics, specifically researcher Gerald Pollack's documentation of exclusion zone, or EZ, water. Both describe cellular water as an active, structured medium. The cell maintains its steep, life-sustaining ion gradients through the physics of water exclusion and selective adsorption. The energetic drain of continuous membrane pumps is eliminated from the cellular equation. If the cell is a solid-state system that does not require ATP to fuel membrane pumps, we have to determine what purpose that energy actually serves. In Ling's model, ATP functions as an electron-withdrawing cardinal adsorbent. An electron-withdrawing cardinal adsorbent is a regulatory molecule that alters the electrical properties and physical shape of the proteins it binds to. ATP functions as a structural pin. Binding to a specific site on a protein, it locks the chaotic lattice into a rigid, highly ordered grid. This binding induces the exact spatial and electronic configuration required for the extended proteins to maintain their grip on the polarized water multilayers and keep the potassium binding sites active. Dr. Mei-Wan Ho expanded on this thermodynamic state, describing the ordered liquid crystalline water matrix as a state of quantum coherence. In this biological context, quantum coherence means the entire water and protein network operates as a single unified system, allowing instantaneous electrodynamic communication across the cell. The massive energy budget of the cell is spent entirely on structure. ATP serves as the lock that holds the matrix in place, preserving the organism's high-energy, low-entropy physical state. The structural models developed by Ling and Ho require a bioenergetic engine to function. Biologist Peat provided the metabolic framework that governs the stability of this architecture. Peat established that maintaining a highly ordered, quantum coherent state against constant thermal decay demands maximum, highly efficient oxidative metabolism from the cell. This metabolic engine relies on specific biochemical governors. Robust thyroid function and high carbon dioxide production indicate cellular vitality, maximizing the ATP necessary to hold the protein lattice together. When metabolism falters, the structure degrades. Peat identified polyunsaturated fatty acids, or PUFAs, as the primary entropic stressor driving this degradation. Polyunsaturated fatty acids are a type of dietary fat characterized by multiple double bonds in their chemical structure, making them highly unstable and reactive. PUFAs undergo rapid peroxidation in the warm cell, fracturing and ejecting reactive aldehydes that strike and warp the protein lattice. Losing its electrical grip, the polarized water immediately dissolves into chaotic free water, and external sodium rushes into the disorganized space. Compounding this localized structural damage, PUFAs directly suppress the thyroid gland, destroying the cell's systemic ability to generate the ATP it needs to repair the broken lattice. The continuous introduction of PUFAs initiates a cascading chemical unraveling. Without the energy to hold its shape, the cell falls from a low-entropy living state into complete chaotic metabolic dysfunction. This model characterizes aging as a systemic surrender to entropy, the steady, physical decline of the cell's ordered state. The macroscopic symptoms of aging, such as mitochondrial dysfunction and the loss of proteostasis, are the physical manifestations of a collapsing protein-water matrix. Proteostasis is the process by which cells maintain their proteins in the correct physical shape and concentration. This thermodynamic lens reframes cancer as well. Malignant transformation is primarily a severe energetic disruption of the cell's structure, rather than an exclusively genetic event. Cancer cells typically exhibit the Warburg effect. The Warburg effect describes how cancer cells switch from efficient oxygen-based energy production to highly inefficient fermentation. This anaerobic shift is a low-energy fallback initiated when the cell can no longer sustain the oxidative metabolism required to hold its structure. Nuclear magnetic resonance studies verify this physical collapse. Scans of malignant tissues consistently show a profound increase in chaotic, highly entropic free water, proving that the structured exclusion zones have dissolved. Disease states and cellular senescence are defined by a loss of physical coherence. The cell loses its shape because it lacks the metabolic energy necessary to lock its proteins and water into place. Therapeutic strategies derived from the solid-state model shift the clinical focus toward restoring systemic thermodynamic order. One approach is orthomolecular hydration. This involves using cosmotropic osmolytes to stabilize the intracellular environment. Cosmotropic osmolytes are specific molecules that encourage water molecules to organize into rigid, highly ordered structures. This intervention artificially forces the chaotic bulk phase water back into a low-entropy state. Peat outlines parallel biochemical interventions to maximize energy flow. This requires removing reactive PUFAs to protect the matrix from chemical damage, while fueling the cell with highly stable saturated fats and simple sugars to ensure robust thyroid support. By restoring ATP flow and supporting the water structure, the cell transitions away from high-entropy disorder, rebuilding its internal architecture. Replacing the concept of the cell as a dilute osmotic bag with a quantum-coherent solid-state model grounds cellular function in the laws of thermodynamics. It offers a clear physical framework for sustaining metabolism and preserving biological structure.